Welcome to part 2 of the Sense and Perception Narrated PowerPoint. Here are a couple of learning objectives for our vision section. Identify the key structures of the eye and the role they play in vision. Summarize how the eye and visual cortex work together to sense and perceive the visual stimuli in the environment, including processing color, shape, depth, and motion. One of the basic things to understand about auditory and visual stimuli is that this stimuli comes in waves and wavelengths. So this slide is showing you the different parts of a wave. First, we have the top peak on each of these. There's two waves that you're seeing there. There is a peak, which is the very top, and then there is the trough, which is the lowest point of the wave. The wave length is the distance between two peaks, and the amplitude is the distance between the peak and the trough. So amplitude, the height of the wave from top to bottom, and the wavelength is the distance between peaks. This slide has an image of the electromagnetic spectrum. This is the range that light waves occur and sound waves occur. If you'll notice, there's the little section that says visible spectrum. So we can only see a small portion of the light waves that exist. Here is another view of the electromagnetic spectrum. In this view, we see how the different lengths and frequencies of the waves correspond to sound or color. On the first example, we see a blue wave. This blue wave is showing that it has a short wavelength. You see the distance between peaks, and it is high frequency, meaning that it is occurring more often. This particular type of short wavelength with high frequency is associated with bluish colors and high-pitched sounds. If you look over where the gold or yellow looking wavelength is, you'll see that it has great amplitude. That height from the peak to the trough is large, and it also has a little bit of a longer wavelength. So great amplitude is associated with bright colors and loud sounds. Looking down at the red wave that we see, we can see that it is a long wavelength with low frequency. So the distance between peaks is far, and the height, the distance between the trough and the peak, is low. That is associated with reddish colors and low-pitched sounds. The greenish looking wave that we see is saying that this is a small amplitude wave, meaning the distance from the peak to the trough is low, is a small distance, and this is associated with dull colors or soft sounds. Next, we're going to talk about the structures of the eye. We're going to start off just with a few definitions of some of the parts of the eye, starting with the cornea. The transparent protective window, it refracts or bends light due to its curved shape. It has a primary role of helping with focusing. The iris is that colored part of the eye. So if you have blue, brown, green eyes, that's the iris. The pupil is a dark hole in the center of the iris. Opening depends on amount of light in the environment. So basically, if you go into a dark theater, your pupil is going to open up to try to let in as much light as possible. When you step out into a brightly lit area, your pupil is going to close up to let only a little bit of light in, because that's all that's needed. The lens focuses light by changing shape. This particular structure is important with helping us to focus. It can stretch out or become thicker or thinner, and that process is called accommodation, the process of the lens focusing light by changing its own thickness. Here we have an illustration of how the eye works. you'll notice the picture of the camera. The way that a camera works is it takes the image and it flips it upside down, as you can see in the illustration. So our eye basically does the same thing. We're going to see the image of the tree. It's going to pass through our cornea, which is that outer layer. It's going to go through the pupil. Then it's going to pass through the lens, which is that kind of grayish oblong looking part. And the image is going to be projected onto the back part of the eye called the fovea. That fovea holds a bunch of different cells, your rods and cones, that are going to help you to process that information. The optic nerve is in the back of the eye, and it actually creates a blind spot in your eye. And in a moment, I'll talk to you about how to determine where your blind spot is. The retina is the back, those cells along the back where all your rods and cones are, they're along the retina. That fovea is one particular area of the retina. So just to recap, the light waves enter the eye through the cornea. The cornea focuses light waves entering the eye. The light crosses the lens, which comes in through the pupil, then it crosses the lens, which focuses the image on the fovea, which is a part of the retina. Photoreceptor cells in the fovea, called cones and rods, when activated, begin the process of sending that information up to your visual cortex. So the sensation part is when these photoreceptors are activated, when these cones and rods are activated. And then the perception part will be once it gets up into your visual cortex. Here is another image of light waves coming into the eye. And here is a close-up of the basic cells of the eye. Here you'll see an image where the light waves are coming in. Those light waves are going to go back and stimulate those rods and cones. The cones are the green ones and the rods are the blue ones. These other, the ganglion cells and the bipolar cells, this is just the structure. There's a chemical process that goes on. You guys don't need to know all that, but you do need to know what the rods and the cones do, and that the rods and cones are existing in the retina. On this slide, we're going to talk about the retina and the rods and the cones. The retina is the part of the eye that converts the electromagnetic energy of light to electrical impulses for transmission to the brain. So this retina is that part in the back of the eye. So this is the area where the rods and cones are located. Rods are thin cylindrical receptor cells that are highly sensitive to light, good for dark and peripheral vision. So interestingly, these types of cells are activated like when you're in a movie theater or in a dark room. They work on your peripheral vision. Now, if you want to learn about peripheral vision, that is the vision that is off to the side. Normally, if we keep our head straight, take your hand and extend it out in front of you. Then move your hand, keep your head straight, but move your arm all the way out to the side and find the place that you cannot see your arm anymore without moving your head. That's your peripheral vision, that vision that's off to the side. Your rods are very active with this type of vision. They are also very good in the dark. Interestingly, if you go into a dark room, the next time you have that experience, you walk into a dark room, try to use your peripheral vision. Try to kind of look off to the side instead of straight forward. And by using that peripheral vision, you may actually have better vision in the dark. The cones are cone-shaped receptor cells responsible for sharp focus and color perception, particularly in bright light. They are concentrated in the fovea. So the fovea is more heavily laden with cones than it is rods. And these particular cells are really important for the sharp detail. On this slide, I'm going to teach you how to find your blind spot where the optic nerve comes in. So what you will need to do is you're going to get a piece of paper. You're going to fold your piece of paper in half, and then on one side, draw a sun. On the other side, draw a happy face. Or you can draw a bee and a flower or anything you want, but you need to have two little images, just like the ones that I have on the screen here. What you're going to do next is you're going to look at the image on the right while closing your right eye. You're going to move that paper away from your eye until the image on the left disappears. So once again, when looking at the sun, you would close your right eye and move the paper slowly away from your face until the smiley face disappears. That would be your blind spot. Then you would do the same thing on the other side. You're going to close your left eye and focus on the smiley face. Then you're going to move the paper slowly away while focusing on that smiley face. You have to make sure to concentrate on the image and use your peripheral vision to see when the image disappears, when the other image disappears. So when you're concentrating on the smiley face, you will use your peripheral vision to see when the sun disappears. That will show you where the optic nerve is causing a blind spot in your vision. You can also try this by moving your computer. I did notice that it worked. When I picked up my laptop and moved it away, I could make one of the images disappear. I could find where my optic nerve was coming through on either eye by moving my computer. Of course, this is only going to work if you have a laptop. I don't think it's going to work with a cell phone because I think the images would be too close together. Nearsightedness and farsightedness. We've all heard these terms, but what the heck do they mean? So in our normal eye, the light comes in through the eye and it is projected to the fovea. So the light image ends up stopping right there at the appropriate place on the back of your retina in the fovea area. If somebody is nearsighted, what happens is the light waves come in, but they stop before they reach the back of the fovea. So the ideal image would be being projected where we see the kind of little twist there in our yellow light beam. Right there is where the beam would be projected for a nearsighted person. So the image back on the retina would end up being distorted or blurry because the ideal image is being projected too far forward in the eye. A farsighted person would have that image being projected too far back. So instead of falling short like nearsighted, the ideal image would be projected at a point that is beyond where the retina is. So once again, it ends up blurry because it is not being projected in the ideal fashion. So that's basically how nearsightedness and farsightedness work in relation to the image coming into the eye. Processing the visual message. Sensory input from light hitting the eye is then sent via the optic nerve to your visual cortex. In the visual cortex of the brain, there are specialized areas called feature detectors. In these feature detection areas, there are cells that are specifically triggered by shapes. For example, anything that resembles a face will be triggered by feature detection that is specifically activated by the shape of a face. Our example of prosopagnosia with our lady that used to be a firefighter, she had damage to this area of her brain. So even though her vision was fine, when it came to the perception, she could not process that information because those neurons in that area were damaged. There are particular shapes or patterns that specific areas of the brain in your optic area, in your visual cortex, will respond to. There are two specific theories about how we see color that we are going to go over in our class. This first one is called the trichromatic theory of color vision. It suggests that there are three kinds of cones in the retina, each of which respond primarily to a specific range of wavelengths. Additional evidence from some types of color deficiency or color blindness support this particular theory. According to the trichromatic theory of color vision, there are three types of cones. And remember, cones are the cells that are important for color detection and detail. The three kinds are red, which respond to long wavelengths, green, which respond to medium wavelengths, and blue, which respond to short wavelengths. We kind of went over this when we were talking about the different types of waves. Any visible color can be produced by mixing the three primary colors. And the way that this theory is supported by color blindness is that this theory suggests that color blindness occurs when one of these cones, the red, green, or blue, is not functioning. Therefore, you are limited in the types of colors that you are able to see. Our second theory of color vision is called opponent process theory of color vision. Receptor cells are linked in pairs working in opposition to each other. Basically, the cells are paired in blue, yellow, green, red, black, and white. And this explains the effect of having an after image. After images occur because activity in the retina continues even when you are no longer staring at the original picture. So the idea behind it is that, say you're staring at something with a lot of blue and you close your eyes, the color that it's linked with is yellow. When you close your eye, you will see the yellow because the blue receptors are fatigued. Same thing if you were staring at green and you shut your eyes, those green receptors are fatigued and you would see red. So that is kind of the basis of this, and our next slide is going to give you the opportunity to view an after image. On this slide, what you should do is stare at the black dot in the middle of the flag. So you're going to start staring now. So just keep staring, don't look away, stare, stare, stare. We want to stare for about 30 seconds. Then close your eyes and note what colors that you're seeing on that flag. You should be seeing red, white, and blue. The next sense that we're going to be talking about is your hearing. Sensing sound. Sound is the movement of air molecules brought about by a source of vibration. Your eardrum is part of the ear that vibrates when the sound waves hit it. The vibrations are then transferred into the middle ear, containing three bones, the hammer, anvil, and stirrup. And the oval window into the inner ear also vibrates. We will be taking a look at these structures in just a minute. In addition, I want to mention that your inner ear is also very important for your balance, and we will be taking a look at that as well. Let's take a look at a few of the structures of the inner ear. The inner ear changes the sound vibrations into a form transmittable to the brain. The cochlea, which is a coiled tube in the inner ear filled with fluid that vibrates in response to sound. The basilar membrane inside the cochlea contains sense receptors for sound, and the hair cells or cilia are tiny cells covering the basilar membrane that, when bent by vibrations entering the cochlea, transmit neural messages to the brain. We're going to go ahead and take a look at an image of or an illustration of the ear, and take a look at these structures a little more closely. When we hear a sound, the sound is funneled into our ear canal by the pinna, which is the floppy part on the outside of our head. That funnels the sound through the auditory canal or the ear canal, and next it vibrates the eardrum. When the eardrum vibrates, it also vibrates the hammer, anvil, and stirrup. If you'll notice, the stirrup is in contact with the cochlea, which is the blue structure. The top part of this blue structure that's all loopy, those are your semicircular canals, and we're going to talk about those in a few minutes. That's your vestibular system, which is important for balance. The lower part that's all curled up like a snail, that is the cochlea. Inside of the cochlea is where our basilar membrane is. The yellow structures that you see are the auditory nerves. So those little hairs that we talked about on our last slide that are inside the cochlea, those are going to vibrate and transmit information to the auditory nerve, where it's going to be chemically altered so that an electrical impulse can be sent up to the auditory center of your brain. This is a close-up and a cross-section of the cochlea. Where the image says number 2, that structure, the kind of pinkish structure that is inside and kind of looping around, that is your basilar membrane. You can see an arrow kind of pointing to that. Attached to that basilar membrane are the hair cells, which is the little lined area. Those are the cilia that vibrate in response to sound. The illustration has high and low written on it. What that is indicating is that at one end of the cochlea on the basilar membrane is thicker than the other end. The area where it says low is the thin area of the basilar membrane. The area where it says high is the thicker area of the basilar membrane. It's thought that these different areas vibrate with the sound in different ways. Let's take a minute to recap about the physical aspects of sound. As we recall from an earlier slide, we have sound waves and light waves that come in, and they have different amplitudes and frequency. So when we're talking about sound, frequency, which is the number of wave cycles that occur in a second, is important for pitch. That's the characteristic that makes a sound seem high or low. So that has to do with the frequency. The frequency is that distance between peaks of the waves. Next is amplitude. Amplitude is a wave pattern that helps in distinguishing loud and soft sounds. So the amplitude is going to be the distance between the peak and the valley. So decibels are how we measure this. The decibels range between the strongest and weakest sounds that can be heard. We're going to look at 2 different theories of sound. There are others, but these are 2 of the main theories. The place theory of hearing. Different areas of the basilar membrane respond to different frequency. It provides an explanation for the sensing of high frequency sounds. So for the place theory is indicating that different spots on the cochlea vibrate according to different vibrations or sound waves that they encounter. So the whole structure is not vibrating at one time, just small areas that are associated with the particular frequency of the waves that are coming in. The second theory, the frequency theory of hearing, the entire basilar membrane acts like a microphone vibrating as a whole in response to a sound. This explains what happens when low frequency sounds are encountered. So the frequency theory is saying that the entire basilar membrane vibrates. Both of these have a little bit of a shortcoming, and most of the theories that we talk about in psychology explain part of a phenomenon, but not the entire thing. So with the frequency theory it does not necessarily explain very high pitch sounds because those are very fast vibrations, and it is thought that the cochlea may not be able to vibrate fast enough, the basilar membrane. And for the place theory, this is based on those different thicknesses that were mentioned earlier along the cochlea, along the basilar membrane. So this theory also falls short as it does not really account well for low frequency sounds. Another important system within the inner ear is the vestibular system. I pointed out those little loop-de-loops to you earlier on a previous slide. So the vestibular system is formed by the semicircular canals. The vestibular system is an important system for your balance. It is how you can understand that you're standing upright. It's also one of the systems involved when you end up getting car sick. So semicircular canals, there's 3 tube-like structures of the inner ear containing fluid that sloshes through them when the head moves, signaling rotation or angular movement to the brain. So when you see a little kid who's spinning around, they spin around, and all of a sudden they're all dizzy and wobbly. That has to do with that fluid moving through the semicircular canals. Within the fluid in the semicircular canals are little things called otoliths. Otoliths are tiny motion-sensitive crystals in the semicircular canal. So basically, when the little kid is spinning around, they're getting those otoliths moving and moving and moving, and when they stop, the otoliths are continuing to move, creating that sensation of being dizzy. These otoliths also help you sense forward and backward movement, up and down motion, as well as the pull of gravity. Now, these otoliths basically depend on gravity in order to work properly. When you are stopped, they're kind of resting in the bottoms of the canals. When you move, they move a little bit. They help you sense your balance. Interestingly, astronauts often get very sick when they go into space, and one of the reasons why they get sick is because there is no gravity. When there is no gravity, these otoliths cannot do their job properly. They can't just sit there in the bottom of the canal when all is well. Suddenly, they're just kind of floating around in the ears, and the astronauts end up getting very sick, throwing up, and having severe motion sickness until they adapt to the environment of no gravity. Here is a nice image of the semicircular canal. You can see on one side of the slide the cochlea, and we know that our basilar membrane is inside of that cochlea. And then we see these loop-de-loops, which are the semicircular canals. You don't need to know the names of the different canals or any of that. You just need to know that these semicircular canals are very important to balance. They're part of your vestibular system, and that there are otoliths inside there that are moving within the fluid that help determine your balance.